This is podcast number 37. Okay, so we talked about glycolysis, starting with a glucose molecule, ending up with two pyruvic acid molecules, two 3-carbon molecules. We're going to further break that down, further oxidize that in this podcast, talking about the citric acid cycle. It's also called the Krebs cycle, after the scientists who discovered it. It's also called the TCA cycle, tricarboxylic acid cycle. Okay, so at the end of glycolysis, we end up with two pyruvic acids. Those two pyruvic acids are going to have two CO2s removed from them, and their two carbon groups are going to be added to a coenzyme. So a coenzyme is not the same as an enzyme, but it's another kind of compound that helps an enzyme do its job. This is called coenzyme A, and those two carbons are attached to coenzyme A. This is getting the pyruvate ready to be oxidized further in the TCA cycle to get more energy out of that cycle. This is called stage 2. In our scheme, glycolysis was stage 1. This is stage 2. In the conversions that happen in stage 2, getting to the point where you're going to strip off one of the carbons. So this is a 3-carbon molecule, strip off one, that's released as CO2, and then you end up with 2 carbons left over. In that series of biochemical steps, you reduce 2 more NADs to make 2 NADHs, and you get the 2 acetyl-CoAs ready to go. Now, this diagram I think is a little bit confusing. Like for glycolysis, I drew out a hand-drawn diagram that has what I want you to remember about stage 2, and after we get done talking about stage 2 and stage 3, which is the Krebs cycle, then we will go over that diagram. So don't worry too much about this particular diagram. Just the idea that it's going from a 3-carbon molecule to a 2-carbon molecule, and the carbon that is released as CO2. You're going to breathe out that CO2. Okay. The energy that it's going to be saved in oxidizing that molecule is going to be saved as reduced electron carriers, NADH and FADH2. Okay, so Krebs is trying to figure out what became the citric acid cycle, but he didn't know it was a citric acid cycle, just knew sort of what went in and what came out. So if you started with a 6-carbon molecule called citrate, also called citric acid, if you like Sour Patch Kids, I love Sour Patch Kids. This is one of the first ingredients. It's a sour tasting thing. It's called citric acid because it donates hydrogen ions in solution. And then that goes through a series of biochemical steps. Okay. Every blue arrow here is an enzyme. And then each of these acids here, acid 2, acid 3, acid 4, so on, are things that have been isolated from this pathway. Doesn't really matter what their name is, just what their order is. And then you end up with something called oxaloacetic acid, which is 4 carbons long. Okay. Along the way, NAD gets reduced to NADH. FAD gets reduced to FADH2. NAD and FAD are just 2 different kinds of electron carriers, 2 different kinds of batteries, like a AA and a 9-volt. They're just 2 different kinds of batteries. And we're going to see in the next podcast how the NADH that gets made in glycolysis in stage 1, in stage 2, and in stage 3. This is stage 3. Then all those built up high energy electrons on those electron carriers get used during the last stage. And then at this stage you make GTP instead of ATP. CO2 is given off. That's 2 CO2s are given off. That's how you go from 6 carbons down to 4 carbons. That's where those extra carbons went away to. And those are breathed out when you breathe. Starts at 6 carbons, ends at 4 carbons. CO2 is given off as a waste product. Why is this significant? So we already saw in a signaling pathway how GTP is used instead of ATP. In the G protein subunit, GTP is switched for GDP in order to activate that subunit, switches back to GDP in order to inactivate that subunit. So GTP is used as an energy molecule throughout the body. In the citric acid cycle of animal cells, GTP is made. In plant cells, it's ATP. A lot of times in intro bio books, it's drawn as ATP just because they don't want to talk about GTP and confuse people. But it's another high energy molecule that looks a lot like ATP. It just has guanine as its base instead of adenine. And if you're going to inhibit the citric acid cycle, that's where it happens. Okay, so this is where Krebs started, and that he thought that this was a linear pathway, that you started with this, you ended with this, and you would see this very similar to what you saw in glycolysis. But then they did some experiments that they had trouble making sense out of, and the experiments were they inject rabbits with a certain compound in this pathway, and then they measure what showed up in their urine. And what they expected to happen was that if I start with this, then the rabbit will turn this into something downstream, this one or this one, for example. Or if I start with this one, I'll get this one. But I would not expect that if I start with this one, that I'll end up with that one, because that would be going backwards and according to how they understood it, and that's not what they saw. So start off with a negative control where they don't inject the rabbits with anything and just see can they measure stuff in the urine, and they can. They measure low levels of things in the urine. Then they take acid number 8 and they inject it into the rabbits, and what they get back out is not what they expected. So if you inject this one here, the stuff that comes before that is what shows up in the urine. So that if 8 is being broken down, how is it that it makes these things? Remember, this is an oxidation pathway here, and if electrons are being stripped off, how is it that these things that are earlier in the pathway show up? So that didn't make sense. If they inject an inhibitor right here, it backs up at acid number 6. Okay, but you do see the one before that too. And if they take the last one in the pathway, most of what they get back out is the first thing in the pathway, citric acid, where it starts. So this was hard to figure out until Krebs had the idea that it was actually a cycle, not a linear pathway, but a cycle. So we start with citric acid, rearrange those molecules into those different acids we saw in those experiments. Along the way, you charge up an NAD to make NADH. You do it again. You make one GTP, okay? And then you charge up a different kind of electron carrier called FADH2, and then NADH again. And then at the end, this 4-carbon molecule is added on to a new 2-carbon molecule, which is attached to CoA, and the cycle starts again. So a 2-carbon molecule comes from stage 2. That 2-carbon molecule came from pyruvic acid, which was 3 carbons, released 1 carbon, made 2 carbons, attached it to CoA, and then it adds itself to the citric acid cycle. So if we're thinking back in terms of how much was produced from a glucose molecule, the glucose produced 2 pyruvic acid molecules, which produced 2 acetyl-CoAs, 2 carbons attached to CoA. And so per glucose, you go around this cycle 2 times. This is for 1 2-carbon molecule. You would do it again for the other 2-carbon molecule that ultimately came from glucose. And you don't get a lot of usable energy out of this pathway in the form of ATP or GTP. But what you do get is a lot of reduced molecules. 1, 2, 3 NADHs per 2-carbon molecule and 1 FADH2 per 2-carbon molecule. And you're going to see in the last cycle, stage 4, where you're going to get a lot of that energy that you put into reducing those molecules. Those molecules get oxidized in stage 4 and you'll get that energy back. Okay, so now let's look at the handout and hopefully what is easier to look at for what I want you to know. Okay? So the stage 2 is happening right here. That 3-carbon molecule from pyruvic acid gets added to coenzyme A. CO2 comes off. It becomes a 2-carbon molecule attached to acetyl coenzyme A. The coenzyme A molecule comes off and it joins the 4-carbon molecule, which is oxaloacetic acid at the end of the Krebs cycle, and then becomes citrate. 4 plus 2 is 6, and you become citric acid right at the top of this cycle. Okay? In stage 2, you charge up 1 NADH per 2-carbon molecule, and this happens twice per glucose. So you get 2 NADHs there, and then in the citric acid cycle, these molecules are going to be rearranged many times, and at several places you will charge up other electron carriers. So NADH, you make 1 there, 1 there, 1 there, and then FADH2, you make 1 there, and at a couple places you drop a carbon off, releases CO2 gas. So here you're going to release a carbon that goes from 6 carbons to 5, and then from 5 to 4, release another carbon dioxide. And on this, it shows you making ATP. That'd be true if it was a plant cell. GTP if it's an animal cell. And you go around this stage 3 cycle 2 times per glucose. Okay? All right, stage 2 and stage 3. So, where does this happen in cells? They did an experiment where they took mitochondrial protein, they added this much protein, and then they added the things that are substrates for the citric acid cycle, citric acid, and they said, what is made as a result of that, and how much oxygen does it take to make those things. So if they add mitochondrial protein but they don't give it any substrate for the enzymes, not much is made. But if they add mitochondrial protein and they do give it substrate, lots is made, or lots of oxygen is used, and lots of stuff is made. If you do the same experiments with the nucleus or the cytoplasm, not much is made at all. So the conclusion from this is that the enzymes that carry out the citric acid cycle come from the mitochondria, and they do. And the oxygen consumed part here, oxygen is not actually consumed in the citric acid cycle. It's consumed in the last part, which we haven't talked about yet. And they're just saying, well, if it's consumed in the last part, it's probably indicator of how fast things are going in the citric acid cycle. But here's what you really want to pay attention to. And the idea that this is happening in the mitochondria. So on these handouts, this handout, if I was giving it to you face to face, would be pink with stage 2, stage 3, stage 4, and stage 5, which we haven't talked about yet. It would be pink because mitochondria have pigments in them called cytochromes, which make them pinkish orangish. Okay, and ultimately, you need oxygen in order to do anything on this page, because all of this happens in the mitochondria. What happens here is this sheet would be blue because glycolysis can happen without oxygen, like you turn blue without oxygen. And you can stop at the end of glycolysis if there's no oxygen around. You can stop with pyruvate. If there is oxygen around, that pyruvate now has to cross both of the membranes of the mitochondrion, 2 membranes in the mitochondrion, and get to the middle part of the mitochondrion called the matrix. And that's where stage 3 happens. So stage 2 starts out in the cytoplasm, and then it crosses both mitochondrial membranes, and then that acetyl-CoA is in the matrix inside the innermost compartment of the mitochondrion, and then all of stage 3 happens in the matrix. Okay?